Life is an amazing concept. Many people think living things need to eat, grow, and breathe to be considered alive. But actually, life starts with reproduction. It all begins with small molecules that aren't even alive but can make copies of themselves. This process of reproduction is essential. These molecules form the foundation for all life we see, from tiny bacteria to big animals like us.
When we talk about life, two important molecules come to mind: DNA and RNA. DNA is the blueprint of life, holding all the genetic information. But before DNA, there was RNA, which is simpler. These molecules are special. Although they're not alive, they have the ability to copy themselves. This copying is how life might have started, as these molecules created complex systems over time.
So now let's talk about the role of DNA. It's known as a very sophisticated structure. DNA makes all life unique because it has so much information in it. But before DNA was RNA. RNA comes first, even though it's simpler. It's like the older sibling of DNA. Understanding how RNA started is crucial to knowing how life began on Earth. It's fascinating to think about the origins, right?
Yes, indeed. RNA plays a crucial role because it can copy itself. This makes it special and important for life's beginnings. Have you heard about viruses? They are interesting because they have RNA but aren't fully alive. That's why we say they exist in a gray area between living and non-living. To our listeners, if you're interested in learning more about such topics, please click Join and become a member. Get access to transcripts and support us too.
The ability of RNA to copy itself is very important. People ask how life started, and it mostly centers on RNA. It's the ability of RNA to make copies that really matters. That's the big question scientists are trying to solve. It's about how life began. The way RNA manages to do this is quite special and essential to the foundation of life.
The complexity of modern RNA can be surprising. RNA sequences can be very long. There's a lot happening in those strands. They line up in different orders, which makes it complex. Imagine a sequence of letters, like a secret code. That's what makes each RNA unique. It's incredible how something so small can have such a big impact on life as we know it. We should keep exploring these mysteries and find out more about them.
When scientists study RNA copying, they look at complex processes. These processes need enzymes. Imagine trying to build something very detailed like a puzzle. Scientists try to solve how RNA could start copying itself just by chance. It's difficult to think about how the right pieces come together without help. People wonder how such a random event could happen. It's like trying to find a needle in a haystack.
Yes, exactly. The sequences of RNA are often very large and complex. These sequences aren't simple. They have so many combinations. The chances of them forming just by chance are very small. They are so big that it's hard for us to really understand the numbers. You might compare it to trying to win the lottery, but with many more possibilities and combinations. Finding that right sequence seems almost impossible.
In past studies, scientists looked for RNA models that could work with fewer letters. They aimed to find simpler sequences that could still copy themselves. This search for the smallest working RNA has been long and hard. Every discovery helped them understand more about life. Scientists kept searching for a sequence that could do the job without being so big and complicated. This has been their challenge.
The numbers are so massive that they seem overwhelming. Thinking about how these sequences can form randomly is a huge challenge. It's hard to imagine that such complex things could happen by accident. However, it is a fascinating part of science. Despite the rarity, it was important for scientists to keep looking for ways these sequences could form without relying only on chance. Each step forward builds on what was known before.
Let's begin with the QT45 molecule. It's an important discovery because it shows how life could have started. QT45 is an RNA that only uses 45 letters to copy itself. This discovery is exciting because scientists thought self-copying required much larger RNA. To find a simple RNA like QT45 is a big step in understanding life. It's amazing how something so small has such a big impact.
Yes, the QT45 RNA is special because it changes the probability of life forming by chance. Before QT45, it seemed unlikely that such a small RNA could copy itself. QT45 shows that it is possible. The fact that this RNA can start the process of copying its own sequence with just 45 letters is groundbreaking. This makes it easier to imagine life starting in the early world.
One of QT45's important features is its accuracy. When it copies itself, it gets it right 94.1% of the time. This balance allows the RNA to change and improve over time, which is essential for evolution. QT45 can also start a chain reaction, increasing its numbers quickly. This means it doesn't just make an exact copy, but a mirror copy too, where each letter is matched with another.
This ability to grow fast through chain reactions is important. It helps to expand the number of molecules in a short time. Understanding how QT45 makes mirror copies is key. The mirror copy doesn't look the same as the original, but it complements the original. For instance, if the original RNA has the letter A, the copy will have U. This ability helps in creating diversity more quickly in early life forms.
Understanding the time factor in the replication of molecules is crucial. For QT45, the 72-day cycle is an important discovery. At first, 72 days might seem long, but in an early world without predators, it is actually quite beneficial. This period allows enough time for significant replication without the rush of survival pressures. The consistent repetition during this period sets a foundation for further evolutionary developments.
Yes, considering the long-term impact, this cycle contributes significantly to evolutionary potential. Over millions of years, genetic diversity can arise, allowing life to evolve in complexity. Each small step taken during these cycles accumulates, leading to significant changes. Early on, the molecules had the time and space needed for many mutations, allowing new traits to develop and helping early life forms adapt to their surroundings.
Rapid mutation and adaptation are key to the evolution of more sophisticated systems. Over time, as these molecules replicate, they undergo mutations. These changes are essential because they allow the RNA to become more complex. Through natural selection, advantageous traits are kept, leading to more efficient reproduction methods. This adaptability is the reason life has progressed from simple molecules to more intricate organisms over billions of years.
While theoretical predictions often seem daunting, actual historical processes were gradual and allowed for diversity. The environment played a vital role in this. For instance, early Earth conditions provided the perfect setting for RNA molecules to thrive and diversify. The interaction between the environment and these molecules led to the creation of diverse life forms. Each environmental shift required adaptations, further pushing the boundaries of what these molecules could do.
Let's dive into the recent breakthroughs in understanding life's origins. Scientists wanted to know how molecules could copy themselves. In their studies, they used natural selection to see which molecules could do this best. It's like a test to see which traits were good for survival. The fact that they used evolution to guide this process shows the power of natural selection, even in lab settings.
Exactly, and what's really interesting is the result of these studies. After only 18 rounds of selection, a molecule called QT45 was able to copy itself. This was unexpected and quite exciting. It shows that evolution can work fast, even in controlled experiments. It's like the lab became a mini version of the early Earth, helping us learn how life might have started here.
Thinking about this in terms of DNA complexity is fascinating. Humans have 3 billion letters in their DNA, but QT45 shows that a self-copying molecule doesn't need to be that complex. It only has 45 letters, showing that life's start is simpler than we thought. This emphasizes the idea that non-living elements can create life, given the right conditions and enough time.
Simplifying the models in this way helps scientists get new insights. It reveals that complex life systems may stem from very basic beginnings. Through these experiments, researchers use the core principles of evolution itself. In addition, these simple experimental designs bring significant understanding about life's start, shaking up our previous assumptions and opening new possibilities for future research.
To understand how life began, we must look at the energy needed to form molecules. Energy is key when these molecules begin to form. Without it, the formation doesn't happen. In the early Earth, energy sources like lightning and UV light were very important. They helped to start these chemical reactions. These reactions led to molecules copying themselves, eventually helping life to evolve from non-living matter.
Yes, these energy sources were crucial. Take hydrothermal vents, for example. These are underwater geysers that act as natural reactors. They provide warmth and energy, which helped life start. In these vents, molecules found the energy to bond and form more complex systems. Without such sources, the odds of molecules becoming life forms would have been much lower. These natural wonders played a vital role.
On our young planet, protection for these molecules was also needed. They needed a safe place to stay together without drifting apart. Otherwise, they would have been scattered and lost. In early Earth, certain environments offered this protection. This helped the molecules combine in stable ways, increasing the chance of life forming. It's all about finding the right balance between energy and stability.
Keeping these molecules together was as important as energy. Without confinement, they couldn't evolve. They needed to be in one place to keep reacting and forming new things. These confined spaces allowed for better interactions between molecules. Today, scientists can learn from this by creating controlled environments in labs. This helps them study the origins of life in more detail, gaining better insights.
Join us on this journey by engaging with our content. Like, subscribe, and share this podcast with your friends. Your support helps us continue bringing you fascinating topics about science and life. Thank you for listening, and stay curious.